They probably would not be mad if you're like, I finished this frap experiment, you just give them a tray of frappuccino. Anyway, let's start talking about membrane transport. So before we get started, I want to put everyone's minds at ease because I know there was a lot in this chapter. So not everything from like the videos and from the slides is going to be on the exam. What is going to be covered on the exam is this stuff, which is still a lot, but it's not everything from the chapter. And all of these are types of things that we'll go over like during class time while we're reviewing chapter 10. Those are the same types of things that will be on the exam. Stuff that is part of this chapter, but not part of like our review stuff in class or on the slide will not be on the exam. That being said, if you haven't like watched the videos yet or looked through this yet, they're still helpful to know some of the other stuff as a general concept, or just to be aware that that's a thing that happens, but not to necessarily recall in a great amount of detail for the exam. So anyone, I'll let you write that down or take pictures if you want. I wasn't going to put this slide in, and then I changed my mind. So here it is. All right. So, still going? Yeah, I got you. All right. So that being said, let's talk about chapter 10. So they're just one big like review area for all those topics at the end of the lecture slides. And we will start it today. We might not finish it today, but that's what Monday's for. So let's start talking about the thermodynamics of transport. So first, let's just take a moment to like read this question together, and then I'll let you think about it individually. So this question is asking, will the following transport be non-mediated, passive mediated, or active mediated? So you had to think about a few things. First of all, you had to remember what all those different types of transport are and what they involve. So some of them might involve the use of energy. Some of them don't. Some of them involve molecules going down a certain type of gradient. Some of them involve molecules going up their gradient. And so to be able to answer the next part of this question, we have to be able to consider what's happening with the electrochemical gradient in this cell. So the question says, movement of 1 mole of sodium ions from outside the cell, where the concentration of sodium is 150 millimolar, to inside the cell, where the concentration of sodium is 5 millimolar. And this is happening when the membrane potential is negative 70 millivolts, and the temperature is 37 degrees Celsius. So we have to think about which direction sodium is going. It's going from the outside to the inside. So this should help you consider if they're going up or down a gradient. And recall there are two components of the electrochemical gradient, right? There's the electrical component and the chemical component, so concentration and charge. So think about how you would know all of these things based on the question. And then once you figure out essentially, are these ions going up or down their electrochemical gradient, that should help you figure out what type of transport they will require. So that being said, everybody take a couple minutes to think about that. right, so does anybody want to tell me if the transport is non-mediated, passive mediated, or active mediated? Yeah, go for it.
I know that the resting membrane potential is negative sodium removals, and by what we want to do is keep the sodium outside of the cell at that to maintain that potential. So when there's an impulse, that's when the action potential goes up, and that's when sodium would come in. So I would say that's passive mediated because you're kind of maintaining that membrane potential with ATP hydrolysis, and then once you do get the action potential, that's when everything opens up. It's kind of mediated, and sodium comes in. Is that kind of that?
So I think you are overthinking the question.
Uh,
But you are also, it's true, it is passive mediated, but the reasoning is much more simple. It would help if I turned the pen on. All right. So yeah, it is passive mediated. So let's think about what's going on outside of our cell and what's going on inside of our cell. So we have concentration of sodium ions, and we have membrane potential. So so let's start with the relatively straightforward one. So what is the concentration of sodium ions outside of our cell? Not a trick question. It says so. Yes, it is 150 millimolar. And what is it inside the cell? 5 millimolar. So according to this chemical gradient, or this chemical part of the gradient, it's going to want, sodium ions will naturally want to diffuse down this gradient. So they're going to want to go from the outside to the inside. If you did not tell them to do otherwise, that's what they would want to do, right? So now let's talk about the membrane potential. So when we say the membrane potential is a negative number, I'm actually just going to change this to charge. Oops. So when we talk about the membrane potential being negative, is that from the perspective of the inside of the cell or the outside of the cell? Which part of the cell is negatively charged? Yeah, inside. So the outside is negative, the inside is negative. The outside is positive. If you are a sodium ion and you're positively charged, which direction do you want to go? Exactly, yes. You also want to go towards the inside because sodium ions are going to be attracted through the electrical part of the gradient to go inside. So now if we consider the electrochemical gradient as a whole, conveniently, regardless of the aspect you're looking at, sodium ions are going to want to move towards the inside of the cell. So if the question is asking for the movement of 1 mole of sodium ions to the inside of the cell, so that's great. That's down the gradient, right? So movement to the inside is so movement to the inside of the cell is going down that electrochemical gradient. So does that mean you need energy to make this happen or not? No, yeah. So no energy is required. So of the types of transport that we have, right? We have non-mediated, passive mediated, and active mediated. Which ones of those don't need extra energy? Yeah, so passive mediated is one of them, and the other one is non-mediated, right? So both of these do not require energy, so we know it's one of those two. How do we decide if it's non-mediated or passive mediated? Yeah, exactly. So because sodium ions are charged, they need help. They're not going to want to go straight through the cell membrane. So they're going to need passive mediated, non-mediated, is for only very specific, small, uncharged kinds of molecules, and sodium ions are not one of those. So you don't need energy, so it's not active mediated, but you need some help. It's not requiring energy just to get through the membrane, so that's why it's passive mediated. Yeah. Because we're going down the electrochemical gradient. So if, yeah. For a sodium ion. So sodium ions are positively charged, so they're going to want to go towards negative charge. Well, when the sodium is outside the cell, it is in a relatively negative environment, right? It's passive mediated. So that's what's happening right now. So outside the cell, yes. If you wanted to pump it back out, exactly. That's when you need active mediated, and that's why we have like our sodium potassium pump. Yeah. Yes. So I guess, yeah, potentially, if you're able to change the environment enough such that the membrane potential eventually changes, and this is what happens during an action potential. So like there's a variety of different channels that are opening and closing at various times, and most of those channels are, not all of them, but some of them are like passive mediated, right? And so like they'll open and they'll allow for the movement of the ions down their gradient. And sometimes that means they come into the cell, and sometimes that means they leave the cell. And it just depends on the local environment, basically. Yeah. For what's happening at the time. Yeah. So yeah, for this particular question, there's not enough information to tell you whether 1 mole of ions is too much and would eventually like change the composition of the environment, because there's not enough information there. But you can assume that it will work the way you expect it to, unless you're told otherwise. Yeah. Any more questions about that? All right. So let's talk about some of the principles of selectivity. So everybody take a few minutes and think about some of the methods and strategies and features that transport proteins use to help them selectively transport a particular molecule or ion while excluding other ones. So we'll take maybe 5 minutes for that, and then we'll come together and discuss it. All right, so does anybody want to tell me any of their message strategies or features that they thought of that help transport proteins be selective? Yeah. Charge, yes. Charge is really important. So a lot of times the parts of the transporter that are facing the cargo, the thing that's passing through, are going to help things with one charge pass through, but exclude types of molecules that have the opposite charge. And so going along with that are also other sort of binding characteristics. So this is things like hydrogen bonding, dipole-dipole interactions, van der Waals interactions. So you have a lot of interactions going on between the transporter protein itself and the cargo that it's helping to transport. And when you use things like charge or other types of interactions, you can help to include certain molecules of a certain charge or electron density and exclude other ones. What other things are important besides charge? Yeah. Size. Yeah. So size is one really easy way to use size is to exclude molecules that are bigger than the molecule you want. So it's really easy to imagine if you have, a passageway that's this big and you have a molecule that's that big, it won't fit. It's much harder if you have a passageway that's one size and you have a molecule that's too small. You have to come up with other, more clever ways to exclude a molecule that's smaller, right? What other types of strategies and features? Yeah. Yeah, I would sort of include that in like charge/binding characteristics, but yes, that is a good thought though. Yeah. Yeah, exactly. So conformational changes. So I think we saw a couple of different examples of conformational changes that happened once the cargo sort of entered the transporter. Transporter changed shape around the cargo to then help it leave out the other side. And those are good ways that can help that transporter be specific for one particular molecule that likes to fit in there. Any other ideas? Yeah. So that's important for transport in general, but I wouldn't say that helps it be selective. There's sort of like three big things that we thought about together a lot. So they're charge, size. What's the third thing we often think of when we're thinking about charge and size? Shape. Yeah, there we go. So the molecule has to be the right shape to fit in the particular transporter, right? Which kind of goes along with conformational changes, but you can think of one being conformational changes within the transporter and one being the actual shape of the cargo that fits inside, right? Any other things anybody else came up with? All right, that is also all that I came up with, so we're doing great. So let's start thinking about these two channels, so KCSA and KV. So I guess first we can probably answer the first one altogether. What is being transported by the KCSA and KV channels? The letter K, yes. What does the letter K mean? Potassium, thank you. So everybody take a few minutes and start thinking about structural similarities of these two channels, and then some of the structural and functional differences between those two channels. Okay, so yeah, so let's get started with this. So I guess first, so we already talked about how both of these channels help to transport potassium. Are they both the same type of transporter, meaning do they use active, do they use passive? They're obviously not non-mediated, but does anybody want to tell me if these are active or passive transporters? Do they require energy input like ATP? They are passive. Yes, they do not need another external source of energy. So functionally, they both allow for passive mediated transport, meaning they're allowing potassium to move down its electrochemical gradient, right? So let's start getting into the structural similarities. So what do we got for structural similarities? Yeah. Yes, they are hetero tetramers. They have one central pore or channel. So that's really great examples for quaternary structure, right? So what about their secondary structures? Any similarities there?
They have two long alpha helix, one short alpha helix.
Yes, so for each subunit, has two long and one short alpha helix. Is there any other sort of structural features that they have in common? Like what makes them both potassium transporters? Do they have a feature that they both have that helps them pick out potassium?
Well, one of them have anionic side chains and the other just has like a insert sequence.
I mean, just because it doesn't necessarily mean they're different from each other, I guess. Yeah. I'm not looking for like a specific, there's like a name. Yeah, they are both, I mean, they're both channels, right? So they open to the outside of the cell. The term I'm looking for is selectivity filter. So they both have the same selectivity filter, right, that helps them pick out potassium. So I think we're kind of over similarities. How about some of the differences? Does one of them have extra structural components that the other one doesn't have? Perhaps.
Just kind of
Yeah, exactly. Yeah, so one is voltage gated and one is not. So for the one that's voltage gated, sometimes it's open. What other states can it have besides being open? Closed. Yeah. Is there a third one? Inactivated. Thank you. And when it's and there's a part of it that helps it become inactivated, right? And that's perhaps a structural difference. Anybody remember what piece of it helps to inactivate it? Yeah, so it has 4 additional transmembrane helices. It also has some other stuff. Anybody? Has a T1 domain. And one other thing has the word inactivation in the name. Inactivation frappuccino. Inactivation. Anybody? Yes, the inactivation ball. Yes, thank you.
So that's all for
The voltage gated one. Yeah, so the voltage gated one has all these extra bits and pieces because it has this extra function, whereas it can be in 3 different states: open, closed, or inactivated. So structure, as I love to remind you, structure relates to function. So extra structure, extra function. And why is it important to be voltage gated and let the channel be open, closed, and activated? What does that kind of allow for? What's that? Yeah, so we have this voltage gated channel that can be open or closed or inactivated. Why is it so important to have these kinds of voltage gated channels that respond to electrical changes? What kind of signals do those help propagate? Yeah, exactly. So this is sort of relating to its function. Helps to propagate action potentials. Need more coffee, yeah. Gotta move those inactivation balls. Get some little hammers and start moving them away from the channels. Yeah. All right. Anybody have any other differences, structural or functional? All right. Well, let's end there because you all seem pretty tired. I will just show the next slide because it just has pictures of them. It's not really a question. But this is showing all those structural similarities between them. So it has all the features they have in common. It has these, this sort of top-down view I think is really helpful. So you have these short helices here, S1 through 4. You have the long helices, S5 and S6. And that's the one subunit, that's the blue one. There's 4 subunits, right? And it's a heterotetramer, so they're all the same, and they're arranged around this central pore. And you can see right in the middle of the pore is your little potassium ion. This is like a side view that shows you the same thing, just how it looks if you're looking like right at the membrane. So the membrane is kind of in here. And then this picture just gives you a better idea of the different components of it, and it points out where the selectivity filter is, which is right in the middle where the potassium ion goes. So yeah, so let's leave it there for today. just some general reminders. Your quiz for Chapter 8 is due tonight. Your quiz for Chapter 9 will be due this coming Monday. And your exam on Chapters 6 through 10 that we are almost done going over is next Thursday. So today is Thursday. So on Monday, we'll finish up Chapter 10. It shouldn't take the whole class time. So if anybody has any questions they want to review on anything from Chapters 6 through 10, please come with questions on Monday. I don't have specific review questions I'm doing with you, so if you want to go over something, ask me on Monday, or come to office hours, or email me, or any of those other things. And I was just chatting before class with someone, and I got a request to move the project update due date. So instead of that being due this coming Monday, the 6th, it will be due the following Monday, November 13th, and I will post that on Brightspace. So now I'm done. Yeah.